sort of paper in the field that defined what are the not signs, but the criteria of cancer. How does cancer come about? And we already know from having me in class is that cells divide when they're not supposed to and become a tumor. Okay, so that's generally what happens in cancer. But how does that happen? And this paper was really a landmark, as it showed. It gave a listing of 6 things that have to happen in order for cancer cells to develop from normal cells. And so for about 10 years, it was the go-to paper to sort of understand the major themes of cancer development. And it has always been known that there is a tumor development is a multi-step process. It's a series of genetic changes that has to happen in one cell in order for that cell to become cancerous. This is not you. I'll have my other class before you come here. Related to this is the idea that cancer incidence increases with age. You know that some young people get cancer, but most of the time people who get cancer are older. And that feeds into this, is that you need to have enough time for those genetic alterations or mutations to occur in a cell over time to make that cell cancerous. So there is an age-related increase in incidence. Most cancer happens to older people. And so, like I said, this paper is going to discuss 6 essential alterations that have to happen in order for a cell to become cancerous. 6 alterations that occur in a cell. One. So the first one is that cells must become self-sufficient in growth signals. Now, in the past, in other classes, we've talked about the fact that cells receive signals from the outside. They receive signals about whether they should live or whether they should die. But what happens in cancer is that cancer cells live on and replicate even though they don't get growth signals. They become self-sufficient in telling them their own cells to grow. So normal cells require growth signals in the form of hormones or things called growth factors. So something like epidermal growth factor, which is a small molecule that binds to receptors and turns on pathways that turn on cell division. Normal cells are going to only divide if they have those signals to grow, whereas cancer cells don't need them. How do cancer cells do that? Cancer cells sometimes overexpress growth factors, which then act in an autocrine fashion. What do you think the word autocrine means?
Self.
Same, self, right. So what cancers can do is they can make growth factors that will feed back and turn them on. So they don't need to have signals from somewhere else in the body to tell them to grow. They can overexpress growth factors, which can turn on pathways to turn on cell division. Growth factor receptors can be overexpressed. And what's overexpression? Well, just what it sounds like. Made too much. They're expressed too much. So in many cancers, in fact, in breast cancer, the HER2 receptor, in about 30% of human breast cancer, the HER2 receptor is overexpressed. It's made too much, and so it then is overactive and is going to turn on the cells all the time. And there's a drug that targets this. Does anyone know what this drug is called? It begins with the H. Herceptin. Herceptin is a monoclonal antibody, an antibody that is made to this receptor. And it is given to patients who have too much of this receptor. So this is an antibody to the HER2 receptor. And it works very well in patients that have that genetic alteration. Now, when I say that this receptor is overexpressed in 30% of breast cancers, that means that 70% of breast cancers, this is not involved. So another thing we're going to find out about cancer is that they're not all the same. Breast cancer is not the same from person to person. There are probably 5 or 6 different kinds. So in some breast cancers, 30%, this receptor is very highly overexpressed. And it turns out that this happens in young women who get breast cancer, usually in their 30s or 40s. And it's a very virulent type of breast cancer. This is the worst type of breast cancer you want to get. Most of these people die pretty early. Although this, in the last 10 years, has turned this around. Because now we understand why this breast cancer occurs, because this receptor is high. And if you can turn off this receptor by using this drug, you can help these people. Okay. Another thing to become self-sufficient in growth signals is to have a mutation in the pathways from receptor to the nucleus. And who remembers RAS? I know I talked about RAS with you in some classes. Who remembers where RAS is? And I just told you where it is. RAS is in the pathway from tyrosine kinase receptors to the nucleus in the growth promoting pathways. So you have that binding of a signal to the receptor, tyrosine kinase receptor, it's going to turn on a pathway, and RAS is activated, and then that turns on a bunch of phosphorylations. Is this familiar? You remember this? So RAS is mutated, and it is constitutively on in cancers, meaning it's turned on too much. If it's turned on too much, what does that mean? It's activating the pathway, and it doesn't need that signal to turn on the pathway. You've got a pathway that's maybe 9 components long. If one in the middle is turned on, you don't need the start to be turned on. The pathway is constitutively on. Okay. And very recently, in the last 10 years or so, they found that most cancerous tumors are not just cancer cells, but also other cells from nearby that foster the growth of the cells. So we're going to call that other cells nearby are stimulated to make growth factors. And this is a really new area of cancer research, is how these cells that are nearby the tumor are actually fostering the growth of the tumor itself. So that is, out of this wheel here, one of the first thing that has to happen for cancer cells to develop is the self-sufficiency in growth signals. The next thing I want to talk about is called the insensitivity to anti-growth signals. And so this is mediated by my favorite protein, the retinoblastoma protein called RB. this was first identified in a childhood cancer of the eye, the retina. But then it was later seen that this protein is involved in controlling cell division in all cells. What it does is it is going to respond to signals that tell the cell to stop dividing. It's actually turned on and off by phosphorylation. The retinoblastoma protein has 15 phosphorylation sites. On serine and threonine. And under conditions where the retinoblastoma protein loses those phosphates, we're going to be able to stop the cell cycle. So upon losing phosphates, RB becomes active, and RB is going to bind to something called E2F, which is a transcription factor. What's the transcription factor?
They help mediate transcription.
Yeah. So E2F is a transcription factor that will bind to the promoters of certain genes and turn them on or off. Okay? So RB, though, controls whether or not a cell goes through the cell cycle, in that it's going to control whether or not E2F has activity to cause cells to go through the cell cycle. So remember the cell cycle: mitosis, G1, S phase, and G2. What happens in S phase? DNA is synthesized, yes. What happens in M phase?
Mitosis.
Mitosis. What happens in G1? It grows. And in G2, also grows and also does some other things we'll learn about. But essentially, when does the cell decide whether it's going to go through cell division?
Is it at the end of G1?
G1. Why in G1? Why would it decide there and not in G2? Yep.
Well, it decides before it copies all of the
It
other
decides before it copies its DNA. So right here is where the cell decides whether it should divide. And how does it make that decision? What kinds of things would make a cell divide or not divide? Gotta talk louder, I'm losing my hearing.
Growth factor?
Growth factor, right? So growth factor, so a growth factor might be made because there's a wound and you need more cells. What kinds of things would tell a cell not to grow?
If it's not needed, if you don't need it.
If it's not needed?
If that cell isn't needed in that little area where it's supposed to be dividing, you don't need any more.
If you don't need more of them, what if there's no space? If there's no space, if there's no nutrients, that cell will make the decision here in G1, I'm not going to divide. There's no space for another one of me, so I'm gonna stay where I am. Okay? So this is what the RB tumor suppressor protein does, is it takes those signals that tell a cell to stop dividing, and it happens here in G1. So it turns out that E2F, not EGF, E2F is a transcription factor that turns on genes required for S phase. And when RB is bound to E2F, E2F is inactive. Can't do that. So what I'm trying to say here is that RB, its activity is regulated by phosphorylation. Its phosphorylation is regulated by whether or not a cell should divide. If a cell should divide, RB RB is going to be phosphorylated. If a cell shouldn't divide, RB is going to be unphosphorylated. If RB is unphosphorylated, it binds to E2F and inactivates it. And it causes E2F to not be able to turn on S-phase genes. So what's happening in S-phase? We have DNA replication. In order to have DNA replication, you need to have many, many enzymes turned on to replicate that DNA. So if you stop E2F activity, cells stop in G1. And that is done by RB only if it's unphosphorylated in its active state. Okay. So growth factors do you think that growth factors are going to make RB phosphorylated or unphosphorylated?
Phosphorylated.
Right. Growth factors are going to turn on the activities of CDKs. Remember what CDKs are?
Dependent kinase.
Cyclin-dependent
Yeah,
kinases.
cyclin.
Cyclin-dependent kinases get turned on by growth factors. Cyclin-dependent kinases phosphorylate RB. Okay. If a cell is not supposed to divide, so no space, no nutrients, or other stress, RB is going to be unphosphorylated. And it's going to bind to E2F and turn off E2F activity. Okay. So just as an aside here, this is where my research lies, is what I've been doing the last 15 years, is this conversion. Can we take highly phosphorylated RB that's found in tumors and convert it to the unphosphorylated RB? And that's what I've been doing and having some success with that. So this is, we'll be talking a lot about the RB protein, because it is my favorite. Okay, so a couple more hallmarks. The next one is, number 3, is that cancer cells need to be able to evade apoptosis. Evade means to avoid, right? So apoptosis is cell death that is highly controlled, that is a genetically programmed process, which is different from just smashing a cell with a hammer, okay? If you smash a cell with a hammer, it's dead. But apoptosis is a sort of very controlled cellular suicide, in which the cell turns on pathways to activate cell death in response to some kind of very bad stress or very bad damage. And we talk about damage to a cell. What's the worst kind of damage to a cell you can do generally?
Prevent its growth cycle.
Break the membrane?
Membrane's bad, yeah. What would be worse? DNA damage.
DNA.
If you damage the DNA of a cell, and you damage it so much that it can't be repaired, that cell's going to have to kill itself, or it's going to become cancerous. So DNA damage. There's essentially 2 things that can happen when a cell faces DNA damage. It can go through apoptosis, or it can go through DNA repair. And that's really going to depend upon how bad the damage is. So if the damage is really bad, the cell will kill itself. But if the damage is not so bad, it will activate enzymes to repair the damage. Okay? So what cancer cells do, though, is they evade this mechanism. They get around it. They have damaged DNA, but they don't kill themselves. And that cell then is able to accumulate more mutations and become more cancerous. So cancer cells do not die when they should. One of the ways that this happens that when you have DNA damage, a protein called P53 goes way up in the cell. And this is going to essentially activate RB and stop cell division. So P53 has been named, back when it was discovered, the guardian of the cell. This thing takes over and is going to protect cells from DNA damage such that it's going to stop cell division. And if the DNA damage is too damaging, too bad, if it's too much damage, P53 causes apoptosis. Now, one of the things that happens in cancer is that P53 is mutant. So this guardian of the genome cannot do its job. It cannot make that decision between should I cause apoptosis or should I cause just the cells to stop dividing. So cancer cells are going to avoid apoptosis. They're not going to die when they should. And two or three more, which are very short. Number 4 is limitless, replicative potential. So cancer cells gain the ability to replicate forever. What does that mean? Apoptosis. Right, so they gain the ability to go through cell division even though they are sort of old. There's something called cellular senescence, where animal cells will divide a certain number of times, maybe 50 to 70 times, and then the cell gets old and stops dividing. That senescence is another word for old. So senescence is the idea that cells age, and cells will only divide, like I said, 50 to 60 times, and then they stop dividing. Now, of course, this is going to vary depending on cell type. Epithelial cells that line body cavities are going to divide almost every day, whereas lung, liver cells are only going to divide maybe once a year. But in general, the epithelial cells where cancer arises, there's a certain number of divisions that these cells can go through until they become old. And this doesn't happen in cancer. Somehow the cancer cells, even though they're old, they still replicate by becoming limitless in their potential to replicate. So cancer cells ignore the fact that they're old and they should stop, and they keep dividing. They ignore aging. And so the last two are very sort of quick. 5 is that cancer cells gain the ability to stimulate angiogenesis. Angiogenesis is the stimulation, let's just call it the growth of new blood vessels. So imagine if a tumor arises out of epithelial cell layer and it starts to grow into a mass of a few million cells. That is a non-physiological event. Those cells are often not fed by growth by blood vessels. They're not. This is not a normal thing. So there's no blood vessels going to this tumor, and so there's a point at which the tumor is going to turn on growth factors that cause blood vessels to grow toward it, because those cancer cells want to live, and so they're going to force the tissues around it to try to keep it alive. So the tumor makes VEGF, or VEGF, which stands for vascular endothelial growth factor, and that stimulates the blood vessel growth toward the tumor. And so this is a pretty new and cool, well not new, but in the last 10 or 15 years, some drugs that target angiogenesis have been starting to become useful. Where if you can imagine an adult who has a tumor, if you can take a drug that stops all new blood vessel growth, it should kill off this tumor, right? Because as an adult, you're not constantly making new blood vessels. The ones you have are here. And if you can take a drug that stops new blood vessels from growing, in theory, you should be able to knock out this tumor. And they do work partially well. But usually in cancer, different combinations of drugs are used. And so a lot of people are trying these types of drugs with combinations of other drugs. And so the last thing that tumor cells need to be able to do or have is that they need to be able to invade neighboring tissue and metastasize. Well, what's a metastasize? Metastasis is a growth of cancer cells that have traveled from one primary site to an additional site. So cancer cells need to acquire the ability to be able to break out of the spot that they're in and travel to other places in the body. And so, and it's the metastasis. So metastasize is the verb, meaning to move, but metastasis is the new colony of cells elsewhere in the body. And for the most part, it's the metastasis that is going to lead to death of cancer patients. So for example, breast cancer, a tumor in the breast is not generally going to kill someone, but what happens is it overgrows the breast, travels through the bones, and travels through the bloodstream to other places of the body, like the bones or the brain or the lungs, and that's where it causes its most damage. So metastasis is a huge area of cancer research in that if we can figure out how does a cell, once it's starting to get too big for its primary tumor, how does it grow out and how does it travel to the bloodstream, how does it survive there, how does it set up a new colony somewhere else? If we can make drugs to the metastasis, then you can stop a lot of mortality that happens with cancer. So I think there might be one more. So what I've given you is really an overview of this paper. And again, I don't expect you to learn every detail, but it is a good background paper to start to get your thoughts together as far as some of the research that we're going to be talking about. Okay. So I wanted to show one more thing, a paper example. In case you haven't done this, but and I think most of you have, when you read a paper, there's a way to do this to make it less painful. And that is to read the parts that are going to be easiest to read first, and to ignore the parts that you don't need to read. Okay? So when you see a paper like this, you're going to have, the first part of the paper is called the abstract. And so a lot of you already know this from taking Bio 490, but there's an abstract here, and it's usually a very dense paragraph of the description of the entire paper. Do not read this, because it is filled with jargon, it is very dense, you won't understand it. What I'd rather you do is to immediately skip to the introductory part. Sometimes this is labeled as introduction, sometimes it's not. Here it's not, but you can. This is the abstract, and here is the introduction. This is where you want to start reading. This is what's going to tell you about the research area, why they conducted this study, why they think it's important. And then in some papers, you're going to have the methods. Sometimes methods are right after the introduction, which are here, experimental procedures, sometimes called methods. And that also is not useful for you to read. So that is sometimes, like I said, right after the introduction. Sometimes it's at the end of the paper. But do not read that. You want to skip that unless later on you can go back and look at it. But that is only going to serve to aggravate you. What you want to do is go to the results section, which is going to look something like this. And in the results section is where they start to talk about exactly what they did. Here in the results, we knew this, so we did this experiment, and here it is shown in figure 1. And so in order to understand what they're talking about, you have to look at figure 1. So here in the text, right over here it says figure 1 somewhere, and then you have to look up at the text, at the figure, which is going to have some kind of visual depiction of the results with a figure legend, which is going to tell you what happened in this experiment. Okay? The first couple of papers, I've picked experiments that are very straightforward. However, you will have questions, and make sure that you ask me. And so as you go through the results, it's going to have a paragraph or two about figure 1. So here's the whole description of figure 1, and then it's going to end, and then it's going to start on figure 2, and it goes through that figure by figure until you get to whatever number of figures there are in this paper, 7 or 8. Here's number 6. And then you get to the discussion. This is also not going to be easy for you to read. However, once you have a handle on the introduction and the results, the discussion is something that you could look over. Okay? It's going to put the work of the paper in context of the whole field. And it's going to say why this is important, why we're great, why part of it didn't work, or what we're going to do next. Okay? And that will also be helpful for your presentations. So I just again I will assign A, B's, and C's to this week's paper today, so you can go on Blackboard and get those for Tuesday. And like I said before, if you have any issues at all about paper figures or questions about anything, just let me know. But one more thing. Let me just show you one thing about some of the papers that we're doing next week. They do what's called a cell cycle analysis to see what stage of the cell cycle the cells were in. Okay? This is called cell cycle analysis. And you're going to see these boxes with these curves in them, and let me just tell you what they mean before, because it will help a whole lot. What it does is it has these peaks that looks like this, and it's essentially DNA content of a cell population. How much DNA per cell do these cells have? And what that tells you is if cells are in G1 S phase or G2. Why? Because this is G1, this is S phase, and this is G2M. If you think about the cell cycle, remember what we just said a couple minutes ago. Here's G1, here's S, here's G2. If a cell is in G1, it has one copy of the DNA. If a cell is in G2 or in M, it has 2 copies of the DNA, right? Because it is going to copy its DNA here and make two copies of it. If it has anywhere between 1 copy and 2 copies of the DNA, it's in S phase. So what these pictures are is a depiction of DNA content, of how much DNA you have. And this first peak is the 1N, the cells that have 1N DNA. The second peak is the cells that have 2 nDNA, and anything in between is the S phase. And so in a lot of cases, they'll give you the numbers here. So this is 31% are in G1, 32% are in S phase, and 47% are in G2M. Okay? What we're saying here is they've done some treatment to these cells, and they've looked at how that changes. They've said, okay, we do this treatment, and now we have many more cells in G2M, so somehow the drug is working to cause the cells